Nanopore is a really funky way of sequencing DNA. It's a little hole in a membrane, and the DNA goes through that hole, and as it goes through, in real time, you can detect the code of the DNA. But you can also identify the changes around the DNA. So the things like methylation groups that are stuck onto the DNA, you can then sequence the DNA and understand those changes around the DNA in real time. We're using this technology, the Nanopore sequencers, to work out what sort of brain tumour a patient has. Currently, that process takes several weeks, and it involves us sending tissue away to London. Whereas with Nanopore, we can do this in the operating theatre suites. The block behind me is actually the neurosurgery theatres, and we've actually done it in that suite. It's a very small box that allows you to analyze the tumour very, very quickly, to gather the genetic data that you need in order to classify tumours. So as we've understood more about the biology of brain tumours, we realise that it's those molecular changes that really predict a patient's future. Those are the things that take us a long time currently to identify, but with the pathway that we've developed, we can do that within a couple of hours to get a diagnosis and then get the remaining important molecular diagnostic data over the next day. There's enormous potential to improve patient outcome here. But I think more broadly, the pathway that we've developed could be applied to other tumour sites. So, for example, sarcoma or leukemia, lymphoma, the same sort of technology that we've been working on could be applied to those pathways too. The Nanopore sequencer, it's a small box, it's relatively cheap, it's relatively easy to use, and so it's got the potential to improve that equity of access to genetic testing for patients all over the UK. So, working as quickly as we can to get it safely into clinical practice, we're hoping that over the next few months we'll be able to get it into routine clinical use.